All right. Good afternoon, everyone, or good morning still. Thank you for staying for the last day. So we'll be talking about our recent tool, protein function prediction tool, ProtFun. So we applied graph neural networks and protein sequence large language models for this problem. So as we all know, proteins are the building blocks of living organisms. Understanding their functions will help us to discover the underlying biology in normal conditions, disease conditions. However, protein function prediction, doing this experimentally is labor-intensive, cost-intensive procedure. So because of that, there has been a lot of computational methods developed in this domain to predict protein function. When we say protein function, we usually mean GO terms, gene ontology terms associated with the proteins. So one protein might be associated with hundreds, maybe even thousands of GO terms. So protein function prediction is a multi-label class prediction problem. GO terms have about 40,000 Go ontology right now has about 40,000 terms in 3 sub-ontologies: molecular function, biological process, and cellular component. So there's been a lot of methods in this domain. Some of the methods were single model based methods, relied on just the sequence or the structure, or PPI, protein-protein interaction networks, phylogenetic information, or the literature data. Recently, more integrative approach also emerged, utilizing graph neural networks, large language models. The next talk will tell more about this, but there is also a competition for protein function prediction happening for several years, CAFA. And several teams compete in this competition, utilizing, again, protein language models, sequence similarity, literature mining, etc. So this was kind of a relatively new area for our labs. We're mostly working on multi-omics data, inferring gene regulatory networks, disease-associated genes. We were working on something else, and we kind of the project evolved into this domain. And what we observed was, in terms of the existing methods, some of them were just relied on the sequence similarity, but similar sequence don't always mean similar function, or similar function don't necessarily require high sequence similarity. There are a lot of methods that utilize PPIs, which is great, but especially for less studied organisms, PPI information is limited. And there were also several methods that utilize LLMs, but just using LLM might be able to miss some of the domain and protein family level information. So we tried to put all those together to come up with this tool. My postdoc, Mohammed, worked on this project from the very beginning. So what we try to combine is we utilize protein LLMs to capture sequence level embeddings. We utilize graph attention networks on a protein family network instead of PPIs to learn functional associations. And we also utilize a database InterProScan to extract features across multiple databases related to domain and motif level information. So this is the overall architecture. The preprint will be available in a few days, and it's currently under review at Bioinformatics Advances. So the paper will be available soon. But this is the architecture. I'll go into the details in the couple of slides. But as you can see, what we do is we utilize a pre-trained LLM to extract features from protein sequences. Those features become node features in a protein family network, and we learn node embeddings through graph attention networks. In the bottom, what we do is we start with this binary vectors that represents domain and motif and various informations about proteins. We create a latent representation using MLP, and everything is combined. All those embeddings are combined in the downstream classification task, which is the Go term prediction. So for LLM embeddings, we tried different LLM methods, and NAKH protein LLM gave us the best result, which is one of the LLMs trained on UNIREF 50 data set. Of course, those LLMs don't know anything about the function. They don't use this description of the protein. It's just based on the sequence. So hopefully there were no like data leakage because of that. It's totally self-supervised learning method. So with that, we got sequence level functional signals. Then we built a protein family network utilizing protein family information, domain information, motif information. So if two proteins are in the same protein family, they are connected, or they share a domain, they are connected, or they share the same motif, then they're connected. So with that, we kind of created all possible protein pairs based on their functional relevance. And to be able to use GNNs, we need to start with some initial embedding. Those initial embeddings were the LLM embeddings for the proteins. So those were the node features. So we utilize graph attention networks for this. And this was trained, all these parameters were trained at the very end. So this is end-to-end model. For this binary protein representation, so this is coming from InterProScan, which collects information from 14 databases. So we collected all this information, has this protein binary signature, which tells basically the presence or absence of a particular feature. We created a latent representation of that utilizing MLP. And these two, the output of the GNN and output of this MLP are combined in another MLP to do Go term prediction. Here, the output layer represents the p-values for each of the Go terms, and we trained this for each of the sub-ontology separately, like biological function, molecular process, molecular function, biological processes, and cellular components. We had separate models. And those proteins, I'm going to show you in the database data sets section, those proteins are coming from thousands of different species. Okay, so this is, again, the overall structure that try to capture different parts of useful information. So we utilize 3 benchmark data sets for this project. And for all of those benchmark data sets, we basically captured the results published in the paper. And then we added our own results to the last row of that table, basically. All of those databases, NetGo, DeepGo 0, and DeepGraphGo, they have proteins from thousands of species, and they come with the experimentally verified Go annotations. So we utilize those as the ground truth. And also to make sure that we have a fair comparison, we utilize the same train, validation, and test split in the original papers. So if you go a little deeper to those databases, so you can see number of proteins covered in each of those data sets. It is from 60,000 to 120,000. The first 2 data sets, when they did the split, they utilized the approach that was used in the CAFA. Basically, the train annotations are based on the annotations up until December 2018. So this is more like chronological annotation. Then the validation is from 2019 to 20, and the test is for anything after 2020, basically until October 2020. And for DeepGo 0, the split was based on the sequence similarity. and some statistics about number of proteins and labels per data set. One thing to pay attention is that especially biological processes, the number of labels are pretty high, like it is one order higher than the other two, like around 10,000. So that makes the biological processes prediction even harder problem, and you're going to see that in the results as well. The prediction results are relatively lower in terms of accuracy. Okay. So in terms of how to evaluate those methods, so we relied on three metrics. These are again used in the CAFA competition. There's a CAFA evaluator software. This is like a published tool that we utilize this. So the first one is Fmax, which is basically F1 score, but the highest F1 score that you can get based on different threshold choices. So like you have the precision recall curve, and the highest F1 score will be your Fmax. So that's kind of a single data metric. S-mean is minimum semantic distance. So this is, instead of only comparing like whether this label is present or not in the prediction, here you're comparing the semantic similarity between the true labels and the predicted labels, because even they are not same, if they are close to each other in the Go hierarchy, you don't want to penalize too much. So this similarity is that the difference between the predicted and the true annotations based on the Go hierarchy. For this, lower the better. And the last one is the area under the precision recall curve. Okay. So here's the first results. So what you see is different tools are is in the last row, and we have Fmax, S-mean, and area precision recall curve, and three sub-ontologies, MFO, BPO, and CCO. Again, as I mentioned, if you look at the BPO results, they are lower because there are way more labels to predict. Overall, our result outperformed the baseline and the state-of-the-art methods for all cases except for area under the precision recall curve in the cellular component sub-ontology. Then for the next data set, again, we outperform almost all cases except for two cases. For this third data set, this is coming from a different paper, and they only had Fmax and area under precision recall curve. So we only showed those from those tools because of the limitation of the paper. And again, except for two cases, our method outperformed the other methods. And again, similar things we observe. Usually the performance is still like we're far away from being really accurate, 0.6, 0.7 in terms of Fmax, the maximum F1 score you can get. And for biological processes, it's only 0.3. But still, again, given the complexity of this problem, this is still hopeful. So these were the results based on the different tools, published tools. And we also wanted to look into what component in our architecture is most informative. So we did an ablation study. In this ablation study, we disabled different parts of the components and look at the results. And this is only one of the data sets. I think this was the Go0 data set. So what we observe is, for instance, LLM means we only use LLM. Like we have the protein sequence, you have the LLM, you just feed it to MLP, and this is the result, which is not too bad. As you can see in some of the results, it's the second best result. Biological protein vector, binary protein vectors, the second row doesn't really help too much. But when you add GNN, like if you have the protein family network topology, then you have an improvement. And if you have LLM, but then you also have the biological the binary vectors, that also improves over the LLMs for most of the cases, even without the graph neural network, even without the protein family network. And when you have all of them, for most cases, we had the best result. For some cases, for one case, it's the second best, and I think that's the third best for the last column. Okay. As a conclusion, we propose ProtFun as a multimodal protein function predictor. It combines features from LLMs, features from protein family networks, and also features from 14 different databases through the InterProScan. We show that it outperformed baseline and SODA methods on most of the metrics on benchmark data sets. And ablation studies also support that multimodal integration helps for most of the cases. And we have the source code and the data set freely available on our GitHub page. There's a Jupyter notebook, like you can just run and generate all these results if you like to. And again, the preprint will be available really soon. With that, I would like to thank our funding resources and my group for their amazing work. Thank you.
Hi, great talk. Thank you. I just have a minor question. Maybe I missed that. So when you're building the family network, you said that you add edges when two proteins are in the same family. So what do you mean by they are in the same family?
Yeah, so InterProScan, there is a protein family information. So there's a protein sequence family, and then there's a list of proteins. So if they're in the same family, we connect them. So if they share the same motif, so if they have a common motif, we also connect them. And also if they have a common domain, we also connect them. So it's a very inclusive network. We have lots of connections. And to make it scalable, because it's a very dense network, we did neighborhood sampling, by the way, when we were doing graph neural networks. Yeah.
And for the family network, you're only using it for training, but not inference?
So when we train the, no, during the inference as well, we use them, yeah.
And so when you predict for an unseen protein, you also run InterProScan, and you find its place in the network?
So yeah, our proteins that are in the test, they're also in the network. But when we train, we don't utilize their labels, like their Go terms during the training.
Gotcha.
During the prediction, we're just doing like a forward pass with the trained parameters, and then we get the embeddings. Those embeddings are also done like a forward pass on the MLP, which gives us the p-values for or the softmax values for the Go terms, and based on that we make the prediction.
Gotcha. And in your ablation study, you didn't look at what only using family network gives you?
Yeah, we didn't. Yeah, we could have. Yeah, yeah, that's a good suggestion. So we tried different combinations, but we tried fewer combinations than the reviewer asked us to do more. And then if you were one of our reviewers, probably we would have done that. Yeah.
Okay, thank you. Thank you so much.
Hi, I enjoyed your talk as well. I'm just wondering, so in the ablation studies, you show, if we think of it as a scale of the parts that make up what you're using to make predictions off of, and you're taking each part away to test to see which one is adding or improving the outcome.
Right.
And we know with all of the parts, it seems to work the best. When it comes to the LLM part, which I find the protein LLMs get used a lot. Like, do we know what is coming out of the LLM? Like, what specifically is biologically relevant? Like, why why it's adding?
Yeah, I don't know that why, but most likely, so LLMs are able to capture some of the sequence features, which are maybe already inside in some of those databases, like binary protein vectors, even better than that. Like, let's say those two sequences, they have some similarity, but this similarity is important because these LLMs found out that those certain amino acids are more important than the others based on their training with lots of protein sequences from different species. Maybe they is able to capture this relationship between different amino acids in the sequence. But yeah, this would be really good to see, like what's happening in the LLM output, and that leads to this. So we haven't really went into the interpretability of this model.
Gotcha. Okay. Thank you.
Thank you.